Hi everyone, I'm Zane Keller. I'm a senior at Seton Hall University, and today we're going to talk about genetic modification, genetic engineering, genome restructuring, genome editing, however you want to call it. So first, today we're going to talk about what is it, what's it all about, how are we using it today, and what are we kind of starting to move into. So genetic modification is just adding DNA to an organism. It's very simple. So if we look at the screen on the right, you notice that it says recipe to cookies, gene to protein. That's essentially what it is. The gene, the double helix that stores all of the DNA, is the recipe of what makes up a person, an organism, a living being. And these recipes produce different proteins. Proteins that break down food, proteins for reproduction, proteins that do anything essential to have a living organism live. And these proteins are important because it's what differentiates you and me from an apple, tree, a squirrel, anything else. So if we continue down this article, you notice that this isn't important. It's the process. So essentially you have your gene that you like, and genes are made up of 4 letters, A, C, T, and G, and a certain combination, just like if you're writing a certain word and you change up the sequence of the letters, it becomes a whole new word. That's how DNA works. We are taking the sequence of letters and rearranging them to be able to create something new, adjust something, modify something. So in this picture you can see that they take the red apple, they modify it by taking it apart, looking at what its DNA structure is, and charting it down. Once you have that, all you have to do is insert whatever DNA structure you want. If you have a DNA sequence of ATCG and you want it to be ACTG, you just switch those 2 letters, those 2 sequences, and you can create a whole new item. Currently what we use this genome restructuring for is to look at plants and see whether we can take away some of the diseases, help them survive certain harsh conditions, and we've been able to take certain seeds and make them more durable, more sustainable, more healthy, produce bigger size fruit, vegetables, what have you. But let's look past the basic plant usage of genetic engineering, and this is going to segment us to the next article, which talks about rules for genome editing. Essentially, this article is talking about the genetic modification of pigs, and now we're starting to transition away from the plant specific genetic modification, moving towards animals. Now, as many of you know, when we do scientific experiments, and by we I mean humans, we start with something that has no feelings, such as a plant. We move towards the animal, so that way we can have some sort of test basis for why we should start using them in humans. I'm getting ahead of myself, but essentially what the article talks about is how we want certain criteria, certain rules and regulations for how we can use genome editing. In this series, I will be referring to genome editing sometimes as CRISPR-Cas9. Okay, that is the genome editing tool used to split apart DNA, put in certain segments of DNA, and rejoin it. What we are looking towards is using the genome editing, the CRISPR-Cas9, to treat diseases such as HIV, hepatitis B, or beta thalassemia, which is a blood disorder known as. So Chinese scientists are working on this to try and find the gene that causes this sort of blood disorder, either cut it out and replace it, or just cut it out. Now the implications are that these scientists don't necessarily know what the effects will be. That's a little scary, and that's why it says we need certain regulations. The National Institute for Health and the FDA have strict regulations that affect the U.S., but China doesn't necessarily have to follow these regulations. So what we are using it for now is for plants, but we're starting to move towards using it for humans. And this is when we get into our last segment. And our last segment is, how are we going to be using it in the near future? Well, some UK scientists are trying to fix a problem with human embryos. They are taking the blood disorder that we had talked about in the previous article and applying the genome editing, CRISPR-Cas9, to it in young embryos. But I want to point out something very important that goes back to our regulation talk, and that is that this section right here, right under the video, the embryo would be used for basic research only and cannot legally be studied for more than 2 weeks or implanted in a woman to achieve a pregnancy. So essentially they're taking what they have decided to do is test these genome editing on human embryos. But the results may not come in as well as they anticipate, mostly because they have such a short window to work on the project, and they also can't see the results from the actual birth of a child. To put this into context, so it can be more understandable, think of if you're writing a code for a program, and you've written your code. You know what code was there before. Let's say you know what code was there before, and you've hit run thousands of times. Now you change the code, but you don't hit run. All you can hit is check for debugs. And the problem is that no matter how many non-debugs there may be, and in this case the DNA sequence inserted into these embryos may be fine, it's hard to understand how the results are going to show if they can't see the final product, the final effect. And the other article that just expands on this is that we have these tools now to modify a human being, and in the next few segments we will be talking about how this tool differs from the one in the movies and what are the social implications. How is this going to affect us from an ethical point of view? What kind of government regulation is going to be needed in order to allow such a tool to exist, allow such a product to be out there? And are we going to have the quote-unquote designer babies of the future? Thank you everybody for listening.